Thank you. Thank you, everyone, for joining us for today's research webinar, Local Partnerships, Developing Solutions for Rapid Needs. And with that, it's my pleasure to introduce our EPA presenters, Jatin Mistry and Helen Buse. Jatin Mistry is a life scientist with EPA's Region 6, which serves Arkansas, Louisiana, New Mexico, Oklahoma, Texas, and 66 tribal nations. He started out as a public health fellow in EPA's Office of Research and Development, which provided him the unique opportunity to understand how drinking water research contributes to regulations under the Safe Drinking Water Act. In 2009, he joined EPA's Region 6 office in Dallas, where he currently serves as the Drinking Water State Program Manager for the state of Arkansas and implements drinking water rules in the region. Jatin has a bachelor's degree in microbiology and a bachelor's degree in medical technology, both from the University of Texas at Austin. He holds a master's degree in public health from the Texas A&M School of Public Health. Helen Buse is a microbiologist with EPA's Office of Research and Development in Cincinnati, Ohio. Since joining EPA in 2007, her research has focused on understanding the role of free-living amoebae, biofilms, water quality parameters, and operational and engineering aspects in the growth of Legionella within drinking water systems. Her current work aims to utilize various treatment technologies to decontaminate and maintain drinking water quality within premise plumbing systems. Helen has a PhD in microbiology and immunology from the University of Michigan and a BS in biological sciences from Carnegie Mellon University. And now I will turn it over to Jatin. Thank you.
Thanks, Shar. Appreciate that. And good afternoon from EPA Region 6 in Dallas, Texas. Next slide, please. Thank you, everyone. Today's presentation is entitled Monitoring Chemical and Microbial Water Quality in Recreational Splash Pads. Here in EPA Region 6, we have an opportunity to work with our partners on the research arm of our organization called the Office of Research and Development. And every so often, we have these opportunities, and they called Regional Office of Research and Development Applied Research Programs. And through these programs or initiatives, it allows a regional scientist or a regional employee to work hand in hand with one of our colleagues at our research facilities on a regional need or a regional research questions. And through this project, we were able to submit a project or a proposal on splash pads, and it was funded. And through myself, Jatin Mistry here in Region 6, and my counterpart, Dr. Helen Buse from the Office of Research and Development, we put together a proposal, and it was funded, and we were able to do this project. And the data that we will be sharing today with you, the preliminary data, would not have been possible without our four partners. To our four partners, we're extremely thankful for your dedication and also your collaboration. And through this project, we are learning a lot. So next slide, please. So let's start with the background and project overview. Exactly what is a splash pad? So a splash pad is pretty much a recreational play area that has little or no standing water. And that's what I want to focus on, little no standing water. To a lot of parents and a lot of users, especially young users, that's kind of like the safety thing. It's not like a swimming pool that has a wide body of water and a deep body of water. These splash pads have little or no standing water, and so that's kind of like a safety factor. The chance of drowning is extremely low or rare. And in these splash pads, what we're also seeing is exactly how the water is being utilized in these facilities. So after it's been utilized or it comes up through a spray or a water feature, the water is either drained, and it could be either recycled or drained. If it's drained, it just goes into a sanitary sewer. If it's recycled, it goes into a holding tank. It is treated, going through a sand filter, things like that, and then reused again in the feature. And there's multiple cycles of that. And also, we have a lot of young children and young folks of ages that interact with these features, and it's kind of like you push a button and a water feature is activated, and a lot of people have ownership and say, wow, I'm controlling this. And so for an adult here, when you're pretty much 4 to 5 feet tall, these spray jets that come from the ground, you don't get that much interaction, or you have more of your situational awareness where, oh, there's a splash coming up, I can change my head or change the direction. And if you're a child that's say less than 3 foot tall, you're going to to be right on top of these spray heads, and you may not have that wherewithal to say, oh, it's coming up, I need to move. They may get a big dosage of whatever's coming from the ground. So yes, next please. Here in Region 6, between 2020 and 2023, we've had 3 fatalities, and that's a very concerning factor. And as you can see on the right side of your screen, kind of those news stories associated with these fatalities. And so through this project, we wanted to do something. Next slide, please. So through this ROAR approach, we have a regional need here in Region 6 to maybe understand exactly what's going on with these splash pads. Is there a link between some of the chemical microbiological indicators that may be pointing to some sort of water quality at these splash pads? And then based on these data, could we glean maybe some operating conditions, some better O&M practices, and things that we can do to maintain additional or enhanced public health protection? So that was the ultimate goal of the project. Next slide, please. So for a splash pad, we have to rely on the drinking water distribution system. And a lot of our splash pads here in Region 6 are connected to a municipal water supply. And what we need to watch out here is that's that source water that's feeding the splash pads. And then also from a splash pad operations perspective, we want to make sure that the splash pad operators has communication with that municipality drinking water system, just for situational awareness of what's going on. And in our water systems here in Region 6, we have some that are treated with chlorine, that's just free chlorine. And then we also have some that are disinfected with chloramines, and that's ammonia and chlorine added together. And when you have a chloramine systems on the drinking water distribution side, you pretty much want to watch out for certain things. Make sure that we have a detectable disinfection residual throughout the system. We want to make sure that our chloramines are, we're maintaining the appropriate ratios out there, and we don't have a nitrification issue going on there. And then also too, at time to time, municipalities may need to change the disinfectant. That means conduct a free chlorine conversion from time to time. And the splash pad operator needs to be aware of when these changes occur in the distribution system. So when that incoming water comes to the splash pad, Mr. Operator can kind of make the modifications as necessary. And also for a drinking water distribution system, flushing is a really good practice. A lot of times we have these splash pads that are at the extreme end of a long distribution system with limited users. And so we want to ensure that the water coming to these splash pads is of the best quality. As you can see in the slide here, we have several links here. The state of Texas has a wonderful resource on chloramine optimization. And then also on the flushing part, EPA has some wonderful resources on that. And those links are not clickable now, but on the PDF, they are active links out there. So that kind of sets us up from the regional perspective, why we wanted to do the project and some situational awareness now. Now I'm going to hand it off to my research partner, Dr. Helen Buse, for the research part of it. Dr. Buse?
Thanks, Jatin. So now just to start, give a little bit of background about a pathogen of interest, which is Naegleria fowleri. So members of this Naegleria genus are protozoa, specifically free-living amoeba that can be found in soil, freshwater, sewage, and warm environments like thermal springs. They have been detected on all continents. There are over 45 species identified in this genus, but Naegleria fowleri is the only known human pathogen that causes the rare but fatal disease of the central nervous system called a PAM, or primary amoebic meningoencephalitis. The disease, unfortunately, has a very high mortality rate. There's been about 300 cases worldwide, 100, more than half in the United States in the last 60 years, and only a handful of survivors, I believe about 4. There's an abrupt onset of symptoms within 1 to 7 days after exposure, and occurs in otherwise healthy children and young adults who have had contact with contaminated water. I listed several disease symptoms here, which include headache, fever, vomiting, nausea, and altered mental status, and signs of meningeal inflammation. Usually results in coma, leading to death, usually within days after the onset of symptoms. Naegleria fowleri has 3 life cycles. There is a cyst, which is a non-replicating and non-vegetative, meaning non-feeding form. And these cysts provide the protection from environmental stress like starvation and chlorination. The trophocyte form is vegetative. They're actively feeding and replicating. And the flagellated form is highly mobile, but they're non-vegetative and non-replicating. And the Naegleria fowleri exposure occurs during water activity. The amoeba enter the nasal cavity via inhalation or aspiration, traveling along the olfactory nerve, then to the crepiform plate. It's the bone that separates the nasal cavity and the olfactory bulbs. I don't know if you can see my pointer. It's just this area over here. Naegleria fowleri continues to travel from there to the olfactory bulbs and then finally to the brain. At that point, the amoeba releases cytolytic enzymes, which causes tissue damage. They can then take up the released host cellular contents as nutrients, hence the colloquial name of this pathogen, the brain-eating amoeba. So regarding their occurrence in drinking water systems, they have been detected in groundwater and drinking water wells. The first study cited here collected about 185 well water samples, with 30 or 16% of those samples testing positive. Lasecki et al. Also showed a high detection rate for Naegleria fowleri in their wells, and groundwater samples, about 27%, sampled at various wells throughout the year. This groundwater can be a source of this pathogen. There have been many published studies on contamination in residential systems associated with PAM cases. Marciano Cabral describes the detection of culturable thermophilic amoeba and Naegleria fowleri PCR detection in various samples from 2 homes associated with PAM cases, including bathtub water. Yoder et al. Describes culture and molecular detection of Naegleria fowleri in bathtub and bathroom sink faucets in homes of individuals whose PAM's deaths were associated with use of neti pots for sinus irrigation. And in 2013, there was a PAM case associated with the use of a lawn, like a plastic sheet slide, with water supplied by garden hoses connected to a residential outdoor faucet. And the paper cited here, Cope et al. Describes the culture and molecular detection of Naegleria fowleri in the soil, garden hose, and hose bit samples from the home. So there is also Naegleria fowleri contamination detected in our drinking water distribution systems. The previous study I might just mention, Cope et al. The group also detected Naegleria fowleri in the town's drinking water service lines and fire hydrants, with matching Naegleria fowleri genotypes to the home samples. And since 2014, the state of Louisiana has been conducting a regular monitoring of Naegleria fowleri in their potable water systems. And in November of last year, Ward and Cherton published their study summarizing this monitoring data collected from 2014 to 2018 of raw water, finished water, and from regulatory compliance monitoring sites like the Total Coliform Room, TCR, additional chlorine residual sites, ACR, and maximum residence time sites, MRT sites. About 29% of those samples were Naegleria fowleri positive. I also included some international studies with similar results. In Pakistan, Naegleria fowleri was detected in public water supplies and domestic systems. Nasal cleaning and ablution, which is religious cleansing, were associated with PAM cases there. And in Australia, has case studies of stable and seasonal colonization of Naegleria fowleri in their distribution systems. This list of summarized studies is not exhaustive. There are plenty of other studies describing Naegleria fowleri occurrence throughout our water distribution systems, which is concerning. This is a map from the CDC depicting all PAM case reports for the past 6 decades by state. Almost all of those span the 15 southern and southwestern states that have extended temperatures, severe summers with short, mild winters. With the expanding geographic range beyond those southern states have been observed in the last decade. Naegleria fowleri there have been cases associated with splashes, like we mentioned earlier with those news articles from the previous slide detailing the recent 2020, 2021, and 2023 PAM cases. The asterisk article here is a very detailed clinical case report of the 2021 Texas case. There have also been some PAM cases associated with surfing and recreational facilities. Wei et al. Describes a recent PAM death associated with an indoor surfing center in Taiwan. Naegleria fowleri was detected in the facility, but not in the surfing pool. However, the health department noted facility design deficiencies that were just conducive to cross-contamination of different recreational areas and inadequate chlorine levels. Miko et al. Described as a PAM death associated with an artificial surfing venue in Texas, where Naegleria fowleri was cultured in sediment and water samples at one of the sampling locations on site. This is just another CDC graphic showing those PAM cases by month of illness onset and probable water exposure. So the blue bars represent water exposures from lakes, pools, lakes, ponds, and reservoirs. The orange is canal, ditches, and puddles. The green, rivers and streams, and the red, tap water. Yellow being aquatic venues. The high number of cases in July and August tapering into September is most likely because of higher temperatures. Favorable conditions for amoeba growth, and also seasonal behavior of individuals. People engage in more water activities when they're looking to cool off, right, and enjoy their summers. Just last week, the CDC published this comprehensive report on waterborne disease outbreaks associated with splash pads between 1997 and 2022. They reported 60 outbreaks, more than 10,000 cases. Luckily, no reported deaths. The etiologic agents responsible for the number of outbreaks are listed here in this descending order. So Cryptosporidium was associated with the most outbreaks within splash pads, followed by bacterial pathogens, Shigella and E. coli, and then norovirus and so on. So as Jatin kind of mentioned earlier, kind of like our research questions, we were really focused on whether Naegleria fowleri is more likely to occur in drinking water distribution systems that use chloramines as a secondary disinfectant versus chlorine. We also wanted to determine if treatment system design and type, how they're operated, and how water within a splash pad is managed could impact Naegleria fowleri occurrence and growth. And ultimately, can we correlate Naegleria fowleri contamination with those engineering and operational parameters with other water quality indicators? The overall goal being that we can find a correlation, can it be used to control and prevent Naegleria fowleri occurrence? So just a brief overview of the project team. Again, like Jatin stated, this is a ROAR project. Jatin and I were able to partner with four splash pads located throughout EPA Region 6, which contains the states highlighted by this yellow border, New Mexico, Texas, Oklahoma, Arkansas, and Louisiana. ORD project team members are all located in our Cincinnati-based research facility, which included Dr. Steve Vesper, Vince Gallardo, Josh Steenbach, and our federal contractor, Sarah Oakham, and Devi Sundarabhadevilu. So as I stated earlier, we were fortunate to be able to partner with four splash pads who were interested in our research questions and were willing and able to do all the rigorous sampling. We will be referring to these splash pads as Site A, B, C, and D throughout the rest of this presentation. Each of these four sites utilized treated drinking water from a distribution system. Two utilized water that were treated from surface water sources and two from groundwater. Three of these utilized drew water from the distribution system that used chloramine as a secondary disinfectant, and the fourth splash pad, Site C, drew water from a chlorinated water system. There were 3 sampling events for Site A, 2 for Site B, and 1 for Site C, and 3 for Site D. And you'll see in a later slide that there were a lot of different samples taken, and depending on the analysis, samples had to be preserved and processed a very specific way. So multiple sampling days were needed. There were also sites that resampled, like Site A and Site D. Thus, they have the higher number of sampling events. For Site C, Jatin and I conducted the sampling because the splash pad operator needed the technical support. So we were able to do that sampling in one day. And I just wanted to mention here that all of the splash pads studied recycled their water and used sand filters. This is an overview of our experimental design. As mentioned previously, these splash pads use treated drinking water from their local distribution system, labeled as distributed water in this the lower left-hand corner. This water can also be used to fill the splash pad storage tank, but that's not shown here. Distributed water enters the splash pad, is treated on site, such as with chlorine injection, then goes out to the various splash pad water features. Water that goes down any drain on site is collected in the storage tank, which is then recycled and treated once again on site. The black dotted line here highlights the various water regulatory lanes. In the drinking water distribution system, the water is regulated under the Safe Drinking Water Act, or SDWA, which gives EPA authority to set national-based standards, and the most direct oversight would be by the state drinking water programs. Once the water goes into the splash pad, past the meter, so to speak, water quality may fall under the jurisdiction of state environmental regulators, state, county, and city health departments, or county, city, park departments. For splash pads, water quality criteria and guidance, operational and maintenance practices will draw from various federal, state, and local codes. For this study, we collected a few liters of bulk water from 2 locations, the entry point for distributed water prior to splash pad treatment and from the storage tank. We also collected a water feature composite sample where 5-gallon buckets were placed throughout the splash pad under or near water features to collect about 25 gallons or 90 to 100 liters of water. We also conducted ultrafiltration of 80 liters of the entry point and water feature sample, which I'll go into detail later. Biofilm swabs were also collected from the drain, and 2 surface swabs of a single or different splash pad water feature on site. Several water quality parameters were measured on site: pH, temperature, total dissolved solids, conductivity, and disinfectant residual. Once samples were received at our EPA Cincinnati lab, we measured for turbidity and hardness. We also analyzed the bulk water samples for adenosine triphosphate, which is an indirect measure of active microbial cells. We also measured for total coliforms and E. coli and heterotrophic plate counts. The biofilm samples were also analyzed for heterotrophic plate counts. In the storage tank samples, we analyzed for 36 different fungal species via qPCR. We measured for various disinfection byproducts in the entry point and storage tank samples. For the ultra-filtered entry point and water feature samples, we also analyzed for Naegleria fowleri. That ultra-filter sample was also submitted for metagenomic sequencing to understand microbial community diversity in those samples. We also performed metagenomic sequencing on the other samples listed here, and those were all also analyzed with quantitative PCR for the various microbial targets and the groups listed here. Because data analysis is still ongoing from sequencing and qPCR analysis, I will only be summarizing a subset of this data with a focus on the Naegleria fowleri analysis. This is a summary of the water quality on site. The data is presented as an average of at least 2 values, except for site C, where there was only 1 sampling event. The pH values were generally lower in the splash pads compared to the source water. And as expected, during the sampling time of year, water temperatures were warm, with the low end at 27 degrees Celsius, which is about 81 degrees Fahrenheit, and the high end of 33 degrees Celsius, which is about 91 degrees Fahrenheit. Total dissolved solids consist of inorganic salts and organic matter in water. The EPA secondary maximum contaminant level for taste and odor concerns sets a 500 milligram per liter limit, but there really is no health-based guidance value. We noted all sites had higher TDS levels in the samples after the entry point, with the highest level seen in site B and C. Salinity at levels usually less than 600 milligrams per liter is good quality, typical of drinking water. And similarly to total dissolved solids, salinity was higher in the water feature and storage tank samples, except for the one at site C. Conductivity, which can correlate with chlorine levels, show higher values in the water feature and storage tank samples compared to entry point, which was expected, except for site C, which I'll discuss further why in the next slide. Disinfectant residuals were also measured on site using the splash pad's existing method, or a Hawk SL1000 portable analyzer, and EPA supplied the ChemKey reagents. FC denotes free chlorine, TC total chlorine, MA monochloramine, and NH3 ammonia. At the entry point of the splash pad, for the samples with a red asterisk, as discussed earlier, these splash pads draw their water from distribution systems using chloramine as a secondary disinfectant. So detection of a free chlorine residual at site A and B was unexpected. There was no data for monochloramine at site A, but site B and D had between 1.3 to 2.3 milligrams per liter of chloramine, and site C had 1.8 milligrams of free chlorine, which was expected from the available distribution system water quality data. Each splash pad uses chlorine disinfectant. So as expected, site A and D water feature samples had high free and total chlorine levels. Site B noted bad Hawk reagents, so no data were available. Site C had chlorine disinfection issues for the last half of the 2023 summer season, and the operator was still troubleshooting when we went on site for sampling. So the low chlorine residuals were not unexpected, which also explained that low conductivity levels we saw in the previous slide. And the facility had been closed during the troubleshooting. Free and total chlorine was detected in the storage tank sample as expected, except for site C due to the disinfection system issues here. Heterotrophic plate counts are a measure of just culturable heterotrophs in a water sample. For the white bars representing entry point samples, we saw high levels of HPCs in these samples. Site A had about 7 logs CFU per mil, and we saw between 3 to 4 logs at site B and D. There were no detectable HPCs in the water supplying site C. For the water feature sample represented by the blue bars, only site A and D did we see decreases in HPCs in these samples compared to the entry point. Site B has slightly higher levels, while site C had the highest HPC concentrations in their water feature sample, but was not surprising, like I said, given the distribution system issues they had on site. Storage tank HPC levels represented by the gray bars were variable. We kind of really didn't know what to expect, but figured counts could be high since splash pad drain water goes into the tank, but there's also long residence and disinfectant contact time in those tanks as well. For example, site A had 2 separate samples that had very different HPC levels, resulting in this large error bar in their storage tank sample, while site D had no detectable HPCs in their storage tank water. We also measured total coliform and E. coli in these samples. Only 2 sites were positive, but only in their storage tank samples. Site A had levels right at the detection limit of 2 most probable number, or MPN per 100 mils, and only 1 of those 2 replicate samples were positive. Site C had higher total coliform levels, an average of 668 MPN per 100 mLs across 2 replicates, with 1 replicate also being E. coli positive, but at that limit of detection. These total coliform culture isolates have been whole genome sequenced to taxonomically identify them and to confirm the identity of the E. coli isolate and find out what strain it belongs to. Here are the storage, like I said earlier, the storage tank samples were sent for fungal analysis, mainly qPCR for 36 fungal species, which are summarized in this graph. There were numerous Aspergillus species analyzed, but we combined them under Aspergillus SPP here. Many of these fungal species found in the storage tank samples are frequently found in drinking water. Fungi help the creation of biofilms, which are the main reservoir of pathogenic organisms in drinking water systems, and most likely in these splash pads as well. Site B and C overall had the highest detects of fungal species, including A. alternata and Cladosporium. These 2 groups of fungi occur in large numbers in the outdoor air, where they grow on the surface of leaves. So their detection in these storage tank samples is not totally unexpected. S. chartarum is a highly toxigenic fungus and was detected in site B, but at low levels. Their presence could indicate there is a source of cellulose in the splash pad, and with cellulose being S. chartarum's typical and preferred food source. So here, going on to the Naegleria fowleri results, I wanted to briefly go over the method itself first. And as I mentioned earlier, typically 47 gallons or 100 liters of a water sample is concentrated by ultrafiltration. The ultrafilter contents are then collected, and an aliquot is placed on agar plates that have a lawn of bacteria spread across the surface to serve as a food source. These plates are incubated at a high temperature for 7 days with continuous microscopic monitoring for growth. As the image on the right shows, you are looking for active amoeba growth, consuming the bacteria as they go. At this point, there would only be confirmed thermophilic or high temperature tolerant amoeba growth observed. There are some physical microscopic differences between species of thermophilic free-living amoeba, but Naegleria fowleri determination is dependent on the next step. So this molecular identification and confirmation step. The agar plate contents at this point are collected, nucleic acids are extracted, and a Naegleria fowleri specific PCR assay is performed. This is done whether or not there is active amoeba growth observed on the agar plate. The agar plate is harvested for this analysis. The site and samples I'll list in the first column, and culture and PCR positivity or negativity will be listed in the second and third columns. For site A, there was no detectable Naegleria fowleri in the entry point sample, so the water feeding the splash pad. But the water feature composite sample was positive for thermophilic amoeba growth, but it was negative by PCR. This is an image of the water feature agar plate showing the actively growing trophozoites and some being highlighted with the black arrow. Site A resampled the next month, and both samples were culture and Naegleria fowleri PCR negative. Similarly, Site B also had negative entry point samples, but there was culturable amoeba that were not Naegleria fowleri in the water feature sample. And this is the agar plate for that sample with a cluster of amoeba cells marked with the black arrow. Site C showed the same trend as Site A and B. Only thermophilic amoeba grew from the water feature sample. Site B and C were not resampled since a day after the sampling. Site B closed the splash pad for the season. And Site D, like I mentioned before, was troubleshooting their disinfection system, so the park had not been opened and then remained closed for the season after our sampling. The 2023 sampling at Site D yielded culture and Naegleria fowleri PCR negativity for all of the samples. The second sampling done at this site this past summer did yield similar results where the entry point samples were negative, but the water feature samples had non-Naegleria fowleri thermophilic amoeba present. I want to mention that the agar plates were also submitted for shotgun metagenomic sequencing. Basically, the entire plate was washed and genomic DNA was bulk extracted. And at this point, I wanted to acknowledge Dr. Justin Wright here from Wright Labs in Pennsylvania, a small business owner whose lab performed the sequencing. Dr. Wright also conducted the challenging bioinformatic analysis to pull out amoeba sequences amidst a large background of bacterial and other microbial sequencing from these plated ultrafilter samples. For Site A, this is a pie chart showing the distribution of amoeba sequence annotations. As you can see on the right, 5 amoeba species were identified from those sequences. The top 3 most abundant taxons identified in the Site A sequences were Naegleria fowleri, with about 60% of those sequences, followed by Acanthamoeba palensinensis, and then Acanthamoeba mauritaniensis. I wanted to highlight a potential database limitation here at this time. These Naegleria fowleri sequences could be Naegleria gruberi. A small subset of these sequences were manually checked on the NCBI database and were about 96 to 100% homologous to N. gruberi. The amoeba sequence database used for this analysis, called AmoebaDB, does not yet have a full reference quality genome for Naegleria gruberi, nor many others in the Naegleria species. Some Naegleria fowleri annotated sequences here could match other Naegleria species. For just sequencing analysis in general, we are constantly relying on database updates and high quality reference genomes. So those limitations are acknowledged in our analysis here. For Site B, we saw more amoeba diversity here, 13 listed on the right versus the 5 at Site A. The top 3 most abundant taxons identified here were Acanthamoeba castellani, followed by Acanthamoeba quina, and then Acanthamoeba guariba. There was a small subset of Naegleria fowleri sequences, about 16 found in this sample. For Site C, the top three were Naegleria fowleri, similar to Site A, followed by Acanthamoeba castellani and Acanthamoeba quina. So in the Site D water feature sample during the 2024 sampling year, no Naegleria fowleri sequences were identified, but the top three taxons we found were, again, Acanthamoeba quina, Acanthamoeba castellani, and Acanthamoeba auroriba. Acanthamoeba castellani and quina have also been associated with amoeba keratitis. So these are also considered human pathogens as well. So to go on, some summary points. The small data sets presented here showed no clear trends or correlations between water quality parameters analyzed. For example, with the fungal detections, the levels did not correlate with source or water type, temperature, turbidity, disinfectant residuals. However, like I mentioned earlier, analysis is ongoing for the quantitative PCR of all the samples, as well as the microbial community and diversity analysis for all the sample types. So perhaps principal component analysis may help reveal some trends once all the data analysis is complete. We also found that there's frequency of culturable thermophilic amoeba in water feature samples, and that suggests to us that splash pad conditions are conducive to their occurrence, even in the presence of high chlorine levels. But definitely more sampling and research is needed to understand what the actual frequency of culturability is in these settings, and if there are any public health implications, then if so, under what conditions. The unexpected result from this was that although samples were PCR negative, Naegleria fowleri sequences were present in cultured amoeba samples. And this could highlight differences between using 18S rDNA-based PCR assay, which is what is used in the N. fowleri method for that molecular confirmation step, versus shotgun metagenomic approach for identification and confirmation. Again, this is with the noted taxonomic database of limitations, which applies to all sequencing analysis. So definitely this area needs to be explored some more. So for possible future directions, since Naegleria fowleri testing is costly and it's also time consuming, we are interested in whether there are indicator organisms that can be used for Naegleria fowleri that are easier to culture and sample for. I think that the rapidity of the method is and the ease of monitoring is key here. Now, I just wanted to end with a highlight of some resources for splash pad and splash pad users. For operating and managing splash pads, the CDC has online resources as well as their model aquatic health code, or the MAHC, which provides guidance to help prevent the spread of pathogens, including Naegleria fowleri in water. Some of the steps outlined to protect splash pad users are to maintain an adequate disinfectant in the water. You basically test before opening to the public each day and test every 2 to 4 hours while the facility is open. You want to maintain water turnover times at 30 minutes or less. Some splash pads are motion activated, but if there's no motion for extended periods of time, the water becomes stagnant. There's a loss of disinfectant residual that can occur, and it contributes to the overall degradation of water quality. So water turnover is very important. And operators and managers would need to also inspect their tank regularly, drain, clean, disinfect as needed. And what I list, this is not an exhaustive list. The MAHC provides guidance, and it's updated regularly based on new information. There are resources about how to stay healthy at splash pads. Splash pad users should also visit their state, county, and local public health and park and recreational resources. There could be very facility-specific water quality information available, alerts, recent news, visitation and public health considerations, and so on the local level that users should be aware of before visiting their local water facility. Here's a link for more public health information and exposure-based information about Naegleria fowleri infections. And also, please feel free to reach out to myself or Jatin. If we don't have the answers, we could probably direct you to someone who does. But thank you for your attention and interest in this webinar.
Thank you, Helen, and thank you, Jatin, for this wonderful presentation. It was extremely informative. I just want to start, we're going to start with questions. So if you have any questions, please go ahead and put them into the Q&A box at the bottom of the dashboard. But I did want to start out with this comment that just came in that the CDC's 5th edition of the MAHC will be released on Monday, December 16th. So it sounds like there's a new edition coming out soon for anyone who's interested in that. And then moving on to our questions. The first question we have is, why are chloramines used as a sanitizer? Chloramines is a weak sanitizer and it smells bad while causing eye itchiness. Either Helen or Jatin, can you all maybe address that one?
Hey, Helen, I'll take that one. That's a great question. In the drinking water distribution system, you are correct. It is a weaker disinfectant. Has on the CT tables, the required CT contact time is a lot greater on a chloramine system than a free chlorine. Here in Region 6, we have to have that fine balancing act between maintaining water quality and watching out for disinfection byproducts. What we've seen is with water system changing disinfectants from free chlorine to chloramines, they are able to attack the disinfection byproducts, specifically the TTHMs. So once that rule, the stage 1 and stage 2 came out, we saw a lot of water systems do chlorine conversions, and chloramines has been helping them out. And it's pretty much a disinfectant that these larger municipal systems use that have very long transmission lines.
Thank you, Jatin. This also might be a question for you. The question is, do jurisdictions in Region 6 require splash pads to have chemical feeders for free chlorine in order to maintain the residual? And do they require additional treatment with UV?
That's a great question. Under the state of Texas in Region 6, splash pads are under the Texas Department of State Health Services, and under their regulations, they do have specific guidance on what is necessary to disinfect the water at these splash pads. And we do see there is under state of Texas regulations a mandate for chlorine disinfection.
Thank you. Helen, this question is for you. Do you expect to publish Legionella results for this project? And if so, when would they be published?
No, we did not monitor for Legionella in this study. We were just focused on general, chemical and water quality, and also Naegleria fowleri.
Great. Next question is, are there concerns with discharging splash pad wastewater into surface water bodies and the spread of PAM?
Sorry, Vic, can you say that again?
Sure. Are there concerns with discharging splash pad wastewater into surface water bodies and the spread of PAM?
I think for some of these recycled splash pads, they'll recycle their water, unless you're talking about splash pads that don't. But then in that case, it goes to the sanitary sewer, so that's treated at the wastewater treatment plant. So I don't think that there would ever be a case where a splash pad would directly discharge into a surface water body. But I guess it would depend on the site.
Thanks, Helen. Next question is, did you explore UV inactivation of Naegleria fowleri as a potential additional solution in a barrier approach with chlorine?
I think one of the sites did have a UV disinfection system, but again, I think the local regulation or the guidance is that they have to maintain a certain free chlorine residual. So even though they were using a UV system, they had to have another treatment type. But I don't think a splash pad can just use UV. Jatina, but I may be mistaken.
Correct. UV can use it as a supplement, but you have to have a residual that can be maintained. So the default is a free chlorine.
Yeah, because normally the chlorine levels required are higher than what is supplied by the distribution system. If that's where if they're drawing their water from that is chlorinated and not chloraminated. So they would need chlorine on site if they're drawing water from a chloraminated system.
Thanks. Another question regarding the free chlorine. Is it a requirement for splash pads with drinking water supply to the water features and no storage or disinfection system on site? The commenter says, thanks, we usually do not check free chlorine concentration on splash pads with drinking water supply.
Based on the local municipality and the state regulatory thing, pretty much the guidance from the state would require at least a free chlorine residual being documented to ensure that you have that public health safety. You need that disinfection barrier at these splash pads because as the water coming into these facilities and based on the location, if it's at the extreme end of the water system, the raw water coming in or the treated municipal water may be not maybe at the lower ends of the disinfection residual, and that additional boost is necessary for public health protection.
Thanks. And the question regarding chloramine use, Jatin, I was just asked if we could repeat that answer. So the question was, why would a provider or commercial site use chloramine instead of chlorine, considering chloramine's weakness as a disinfectant?
That's a great question. Again, on the drinking water distribution side, as on a surface water treatment plant, the primary disinfectant is chlorine, but then we have a lot of large surface water systems that add that secondary disinfectant called chloramines, and that's ammonia tied with the chlorine. And the reason why we see this a lot in Region 6 is, yes, it's a weaker disinfectant, but that disinfectant lasts a lot longer. And it's a balancing act that we need to maintain for the disinfection byproduct rule. We have some disinfectants like total trihalomethanes where we have chloroform, dibromomethane, and some other constituents out there. And we want to make sure that we're not exceeding those values that may cause some other public health concern. So an alternative to that is using chloramines, whereby those DBPs can be managed and where we also maintain at least a detectable residual, but we're also keeping an eye on our disinfection byproducts that the system may be creating.
Thank you, Jatin. The next question is, can you talk a little bit about the difference between a splash pad and comparing it to a traditional pool? They said that it might be obvious, but can you all talk about that at all?
Sure. There's just less volume of water in a splash pad. Like Jatin said, there's just basically kids that can run around and interact with water features rather than just like a large body of water like a swimming pool.
Thank you, Helen. Another question specific mostly to Michigan is we have a splash pad in Michigan and their discharge goes to the storm sewer system due to removing that inflow into the wastewater system. Are there any other options for treating?
I'm not familiar with Michigan rules if they have criteria for what can be discharged into the wastewater. If there are, depending on if there's some that have chlorine limits. So if the splash pad has a really high level of chlorine, I would say that might be a concern at the wastewater treatment plant. But otherwise, I don't know of any specific. Like I said, every local, county has their own guidance on those types of rules and regs. So without knowing exactly, is there a specific county or city that we're referring to? We can follow up and look into those for that person.
Thank you, Helen. Yeah, and nothing further than Michigan was provided, but if the participant would like to ask further questions, Jatin and Helen's emails are up here on the screen. The next question is, are there any studies currently being done at other aquatic venues that use special features such as wading pools or special use pools?
I'm not familiar with any in my colleagues here in ORD, but I don't know if there's other groups in academia or elsewhere that are doing those.
Thank you, Helen. And I think, Helen, you had pointed, and Jatin, you all had pointed to some guidance, but there is a question on here of what guidance do water parks commonly use to prevent Naegleria fowleri? Maybe just go to the slide if that includes that. Be helpful. Perfect. All right, thank you, Helen. The next question is, what free chlorine level is necessary to inactivate the cyst form and the trophozoite forms?
Generally, the cyst form is more resistant depending on the pH and the temperature, because those parameters will determine inactivation efficacy. It could be up to maybe 5 to 50 milligrams per liter for cysts, again, depending on the experimental condition. And for trophozoites, they're much more susceptible to chlorination. So again, depending on the temperature and what else is in the what else can be exerting a chlorine demand, anywhere between like 0.5 to 2 milligrams per liter of free chlorine is what I've seen in the literature. But again, it's very situation dependent, and whether or not the trophozoites and the cysts are in the bulk water or if they're within the biofilm, because the disinfectants would have a harder time penetrating the biofilm matrix.
Great. Next question is, can PAM still be detected while source water is chlorine treated and then recycled water from splash pad is UV and chlorine treated while maintaining a safe free chlorine residual? Hopefully I got that. I'm
not sure if it's referring to PAM, if the organism can be detected. PAM is the disease. So they're talking about cases where there is chlorine. I mean, there are some studies where a loss of disinfectant residual is associated with PAM cases. But if they're talking about the method, you can, for example, the PCR method can detect Naegleria fowleri even if the sample was culture negative. But the PCR assay gives no information about the pathogen's cellular status. So if they're injured, dead, or in a non-culturable state. So there could be a few possibilities if Naegleria fowleri could not be cultured in the water sample, but the sample is PCR positive. It could mean the cells are dead or they can't be grown from the sample.
Thanks, Helen. And I know we had a previous question about other aquatic venues, but someone else asked a question regarding any studies with hot tubs and extended water retention time. Anything you're familiar with?
Not specifically about retention time, but you know, like I mentioned earlier in the slide, Naegleria fowleri can be isolated from thermal springs, so in a really warm, and they are tolerant up to like 45 degrees Celsius. So those warm bodies are also a risk.
Thanks, Helen. Next question is, what is the typical operational period of a splash pad?
Hey Vic, I'll take that one. Here in Region 6, we typically see our splash pad operations begin around Memorial Day and go all the way to about Labor Day. And then depending on the temperatures here, as we have been, our fall seasons have been coming up, occurring more in the later part of the year, November, December, then we may see after Labor Day a limited operation like weekend operational hours, things like that.
Thank you, Jatin. Next question is, what would it mean if Naegleria fowleri was detected by PCR but could not be cultured?
So like I had said earlier, yeah, that means that the PCR method used was able to detect Naegleria fowleri DNA. So the PCR doesn't give any information, right, whether the cell is injured, dead, or in a non-culturable state. So and Naegleria fowleri cannot be cultured, but the sample is PCR positive, it could mean that the cells are dead or they can't be grown from the sample. Or that the whatever chlorine residual is there or whatever disinfectant is keeping it under control that would enable it not to be grown from culture, but you can detect it by PCR.
Great. And a similar question is, are there filter systems that direct water through a boiler to kill Naegleria fowleri?
Not that I'm familiar with that splash pads would utilize.
All right. Next question is, and I think there was a similar question earlier. How can you tell the difference between the trophozoites and cysts on the agar plates?
Sure. The trophozoites can be large, about 35 microns in length, and they have a single nucleus with several large vacuoles, and they're very active, and they're very active on the plate, and they can move in a wave-like pattern or like a slug-like form. You can sometimes see pseudopods, which are these arm-like extensions, whereas the cysts are smaller, about up to 15 microns in diameter. They can be round, oval, or polygonal, and they have a double wall structure, and depending on the species, there may be more than one nucleus in the cyst.
Great. Thank you, Helen. And now that we are at the top of the hour, I'll take one last question here. And that is, what type of communities did this study take place at?
Yeah, great question, Vic. Again, this project wouldn't have been a success without our 4 partners. And what we did for these systems, we looked at water systems that were pretty much medium to large size, so greater than 3,300 in population, that what EPA considers a medium-sized water system. And we looked at different sources, surface water, groundwater, and then also the source water using different disinfectants. So yes, so pretty much mid-sized communities, 3,300 and greater.
Great. Thank you so much. Thank you, Helen, and thank you, Jatin. And this is our final webinar for 2024. For those that are on the line right now, we are getting ready to put up our 2025 webinar list here pretty soon on our website, and we will be sending folks out with in the new year. So happy New Year to everyone. Thank you, Helen. Thank you, Jatin, for this great webinar. And with that, we'll go ahead and end.